about the potato bar. So I won't introduce myself except to say, hi, my name is Carrie, and I'm the soybean breeder here. Today, I want to talk about, my research topic is going to be about assessing and maintaining genetic gains in the NDSU soybean breeding program. But before then, just because I have an audience, I don't like to repeat myself a lot. So I'm going to make an announcement. I'm changing my name. I'm taking my husband's last name. So my name will not be Carrie Miranda. It will be Carrie Dottie or Dr. Dottie. And so, you know, part of this is for family reasons. And then part of it's also that having two first names is not actually as cracked up as people think it is. In fact, I would think that at least 30% of people in this room were convinced that my first name was Miranda. So two birds with one stone there. So I will eventually get the process to change your name in North Dakota is quite interesting. I had to like announce it in the newspaper first. So short of the long, it's a unique process, but eventually I'll have a new email address coming. Okay, so first things first, for some of my research to make sense, you need to understand this genotyping platform that I used, but also Agriplex is a sponsor of this event. So I feel obligated to plug their service a little bit. And then also I'm co-creator of a new SNP panel. Yes, I know, another one, a new Soy Community SNP panel. This one is more around 1,000 markers. So the need for this panel is because the most commonly used SNP panels, the 6K and the 50K arrays through Illumina are being retired. So if you didn't know that too, sorry to break that news to you. 50K is already done. You can still purchase the 6K array from Illumina, but by the end of the year, it's being retired. So, and a lot of the reason for this is that the array is produced through Illumina, but then you still need a service lab in order to do the genotyping. And most of that was done by Kijun Song. He designed these SNP arrays and then was also doing the servicing for the sequencing for the community. And between he and David Heighton, who had these services, they're both like, we can't actually do our own research because we're so busy doing this service. So essentially, there was a need in the community to outsource this, as always, right? Let's outsource it to a private company so that we have more free time to do the research that we need to do. So Kijun Song went through the 6K, which is a subset of the 50K SNP panel, and he curated about 1,200 markers that span the genome. And essentially, he just reduced the number of SNPs based on linkage blocks. So in soybean, we have really large linkage blocks, so it was not so difficult to reduce the number. Even 1,200 is arguably more than most of us need to use, especially for genomic prediction purposes. And so these SNPs are evenly spaced throughout the genome, inside and outside of major haplotype blocks. Just for some validation, in case you were worried, there were cutoffs for minor allele frequency, where if it was less than 0.1, it was thrown out. And then they tested the usefulness of these markers in both the Southern and Northern programs, because if you're familiar with that, the Southern and Northern soybean breeding programs are actually genetically different. So they do have different needs. And this panel has a minor allele frequency of 0.29 and 0.33 respectively to the Southern and Northern elite programs. So very useful in my opinion. The trait marker is that's where I came in. I would like to say, hey everybody, I created 88 unique trait markers, but that would be maybe a career's worth of work. So I actually just went through and petitioned the community essentially for them to contribute their markers that they were using most often for this panel. Because what keeps happening with the 50K and the 6K arrays, you have to find associated markers for these traits that you use all the time. Why do I need an associated marker for E1 when I already know the causative SNP for the E1 to E1AS mutation? So I was like, while the community was had this opportunity to contribute, I was like, please, 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 let's include the traits that we actually use the most in the community. And thankfully, people went with that idea. So I was able to collect 88 markers from breeding programs throughout the country. And so quite a few of them are what I would call a perfect marker, where the marker is the exact causative SNP that changes a phenotype. And then in cases where that wasn't available, especially for the diseases, then we have an associated marker. A proxy marker, as Nick said. Actually, I'm really grateful for the student presentations because they did a lot of the background explanation that I absolutely just scoot over in my presentation. So this is just a chart to show the 1,200 markers and how they evenly span the genome. There isn't much need to go into more detail than that. Hopefully you just take my word for it. And then these are the 88 trait markers. They can be categorized into agronomic. So some of the markers might be a little superfluous, flower color. There are still plenty of people that are like, why do you need a marker for flower color? You can just look at it. Sure, I get it. But especially things like pubescence color, stem termination. So if Bob Stupar is on the line, I hope you're hearing this one. Some of these things are, stem termination is definitely something I genotype for heavily. And every time I genotype for one of these traits in my lab, it costs me about $3. Okay, so keep that in mind. And so, especially seed composition, oil, oh my goodness, oil, protein, these are things that we do select for with breeding. These are valuable markers. Flowering time and maturity, this might be more research-based, but it's the kind of research that I do. That was actually the reason why. So Nick showed that there were 3 E genes that we look at. Think about it. So 3 times 3, that's already $9 I'm spending on each line. Then there's some pathology traits, 32, which these do require a bit of a user manual in order to interpret. So there is a publication coming out about how to use these trait markers. And then physiology. IDC tolerance. I contributed these markers from Phil McLean's work here at NDSU. That's 8 markers. So, and his paper pretty convincingly showed that you really need to have all 8 if you really want to understand your IDC score. So again, 8 times 3, that's $24 plus the maturity ones, plus my stem determination. So already I'm talking about per line, well over $30. For the traits that I'm interested in. Oh geez, okay, so these were made by Equiplex. So I'll break it down. They do what's called community pricing. So I told them that breeders and geneticists typically collect leaf tissue from their field studies, maybe in like June, July, because that's the correct stage that you would take leaf tissue samples. So if everyone collectively sends it in at the same time, then we get reduced pricing. So, which I'm sure everyone's like, how much does it cost, Carrie? So I sent in my samples and in a non-community time. I got my results back in 1 month and I paid $8 a sample. If we can get the community involved, that price could go down to about $6, maybe even $5 a sample. And again, like I said, when it comes to the traits alone, I'm already paying $3 per trait. So if you're interested in the traits, this is definitely a bargain. If you're interested in the genomic selection, then this is definitely a bargain compared to what's currently available. Also a huge perk. If you know anything about lab costs, DNA extractions included too. This to me is a huge bottleneck for genotyping. Is actually extracting your own DNA. So I know people who even have that like high throughput DNA extraction robot. What I think you can do 500 samples a day. And the kit alone still costs about $7 a sample. So now for $7 a sample, you can get your DNA extracted and get all your genotyping data. So we are definitely in the age now of where people, when I started school and people were like, genotyping will get cheap, probably to the point where, oh, great, genotyping is cheap. So if you're interested in utilizing this, there's the contact at Agriplex, or you can just ask me, because most likely it's easier to ask me. So I have used this twice already, and my data that I'm going to present has utilized this panel. Okay, so now for the heart of my talk, assessing and maintaining genetic gains in the NDSU soybean breeding program. So I'm an early career scientist. I've been in my position now for a year and a half. I am a breeder, right? I came here, I was hired to be a breeder to release varieties. And then my mentors came up to me and said, well, what's your research contribution? What are you giving back to the community? And I'm like, what are you talking about? I'm making superior soybean varieties. Can't I just be known for that? And they're like, no. You still need to do research too. So what are you going to be known for? So now apparently I have to be known for two things: being the best breeder of soybean in my generation, hopefully, and then also having excellent research. So I find it easier to think of this as two separate entities: the NDSU Soybean Breeding Program, that's obviously going to live on after me, I hope, and then my research program, which I'm now calling the Dottie Lab. So my mission statement for the Dottie Lab is that this research will directly contribute to the creation of high yielding and disease resistant North Dakota cultivars. So the two are separate pieces of work, but they're not mutually exclusive. What's most important in my research lab is that I rely on essentially every single person in this room in order to help me achieve the research goals I need to produce these cultivars. I cannot pretend for 1 second that me as a breeder alone is going to make what's necessary to give superior varieties to farmers. So thank you all, essentially is what I'm saying. And I look forward to our collaborations in the future. So next, this is very general, right? You could still say, Carrie, what's your research? So my first task was identifying a problem. Well, it's pretty easy. We've heard it quite a few times. We've heard it from the Soybean Council, from Stephanie's talk. Soybean is the #1 crop in North Dakota, and it's #4 acres planted in the entire country. But unfortunately, these stats are from the USDA. Notice that 2021 isn't included. That's also because it wasn't published at the time. But also, I think I can exclude 2021 data due to the horrific drought that we had. So if you compare us to other dry land, that's important. That's why Nebraska isn't here. If you compare us to the other dry land, central and northern Midwestern states, we are at the bottom when it comes to yields. Now, there are many reasons for this. So before Hans starts coming up and saying, wait, what do you, I have things to say. I'll acknowledge first its environment, right? Our environment can't, from just the 2 years I've been here, our environment is very unpredictable. We had an early frost in 2020. So if you had a later variety or you planted late in 2020, we had our early frost, I think September 6th, which was crazy, a killing frost September 6th. And so that did have a negative effect on yields. Flooding, I have to usually plant, what do we do, like 17 locations or something like that, because the flooding risk is so high. And then I'm worried about flooding, picking my locations based on flooding risk. And then in 2021, we had a devastating drought. Also, if you look, it's not really fair to compare North Dakota to Illinois. It has nice, beautiful black soil everywhere. yes, the eastern part of the state is very rich. I mean, this is definitely the most fertile area in the Red River Valley. But then as you head out west, that rainfall drops, right, Eric? That rainfall drops. Some parts by Williston, by the Montana border, get about 3 inches of rain per season. So you can see, but they still produce soybean, you know? So you can see why we would have discrepancies between based on the geography of the state. Also things though that we should consider, soybean is a new crop. Soybean's been grown here since 1980 and before, but really acreage has only exponentially increased since 1995. So that's less than 30 years, right? I had to do, if my math is wrong, you can call me out on that. We really don't, we still need time to learn the proper management for this environment. And then as a breeder, I have to ask the question, what about our genetic diversity? We have only been consciously breeding for soybean in this state for 30 years. So we don't have the benefits of some of the other central Midwestern states that have been working on it since the 60s. So this area right here, to me, seems like it could be a jackpot if we can increase genetic diversity, if we can identify that as the problem. So when it comes to genetic diversity, there are known mechanisms that are incredibly important for yield. Notice that I put these together: maturity, season length, stem architecture. So we know, especially in North Dakota, we are limited by that early frost on both ends. Or excuse me, a late frost in the spring and an early frost in the fall. We are limited by, our season length is limited by this. But as Hans said, the earlier you can plant and get as much of that field time in as possible, you're going to see yield gains. However, you also have higher risk. So how can we maximize our yields but reduce our risk without having too long of a maturing line? And so this was actually something that I was interested in during my PhD. And one thing that's been done in other states is that they've transitioned their programs from these long season lengths to shorter season lengths, but then reduced flowering time. Now, there is concern that flowering time might be more influenced by the environment this far north than in other states. And so that's something that needs to be explored as well. But if we do have control over flowering time, decreasing the vegetative stage while fixing stem architecture to maximize yield without lodging could be a potential source of yield gain right there. Also, I always have to appreciate the pathologists. I can work and make a North Dakota variety that yields 80 bushels per acre. But I'll tell you what, if it's susceptible to Phytophthora, that 80 bushels doesn't mean anything because nothing is growing on that plant and nothing will survive. And now we're starting to get pressure from soybean cyst nematode and SDS. So Kyle, if you can believe it, SCN is still only a new threat here. Phytophthora is still one of our worst diseases. And then we also have the physiological issue of iron deficiency chlorosis. So these are our main stressors here. So without these all stacked into new varieties, the yield is not useful. So it's a balance between protecting the yield and increasing it at the same time. And I'm also curious about essentially yield QTLs. Are there mechanisms that are in the background of soybean plants that we don't even really question that could actually lead to yield? So I know that trying to map yield QTLs in soybean has been quite elusive, but Randy Nelson, who was the former germplasm curator of the USDA, I picked his brain before he's like officially retired. And he very convincingly made me think that there are yield QTLs. And so that is something I also want to explore. So just to give you an explanation of the soybean program so my research makes sense, the program is divided up into 4 groups. I have 2 commodity groups, glyphosate tolerance and conventional. Glyphosate tolerance obviously is GM. It's the most commercially successful in the program right now. And then I also have tofu, which is essentially a high protein bean, and then natto, and both of these are specialty food grades. So there is surprisingly more demand for tofu lines. So that could possibly be a quote unquote commodity release, but the natto is certainly specialty and needs a special agreement with a private company. The breeding program includes maturity group 00 and 0. So 00 is the earliest, it's over by Canada, and then zeros are going to be later, central North Dakota and closer to South Dakota. Historically, NDSU, actually I should say NDSU, Ted Helms. Ted Helms was my predecessor. I'm extremely fortunate that he left me with such a flourishing breeding program. And he released 40 varieties in the 30 years that he's been breeding here at NDSU. And then 20 of these, well, so far that I counted, 20 of these are commodity lines. And this is what I used in my analysis. I also have historical data from the USDA uniform trials. So thank you, Clayton, for explaining what those are, because I was not intending to. And then of course I have our own NDSU records. But Clayton did a really excellent job talking about how difficult it is to actually assemble the historical data and then make it useful, because it's cumbersome. And then I also have the pedigree data for all these lines. So first thing, I wanted to look at genetic gains over time. Ted released his first variety in 1994. And I have actually released new varieties just last year in 2021, but I didn't include them here. So Ted's last release variety was in 2020. And so the way I calculated gains was simply take the last the last release variety, subtract it from the first release variety, and then divide it by years in between. So it's not completely accurate because you can see the R² value for this line is not great. And this includes both maturity groups, because actually if I separated them, the data was less interpretable. So this is only preliminary data. It's definitely have some work to do to clean this up. But short of the long, our genetic gains of the entire life of the soybean breeding program is about 0.48 bushels an acre per year. So, and these data are all from the uniform trials. So you might say, neat, Carrie, what does that mean? So for context, let's look at the historical data from a different maturity group that's been around a lot longer. These data are from the Deers' lab. It includes data from their breeding program and also private industry. So this is Ted Helms, and then this is all the soybean breeders in Illinois and Iowa and Indiana. Okay, so a lot of power behind this. Here, the first variety was released in 1925, and the data stops being tracked in 2005. Comparing our gains to maturity group 2's, their yield gains are actually 0.33 bushels per acre. But that's pretty much because what is the denominator in this equation is time. So they've just had more time to increase their yields. So essentially, given our short time and the yield gains that we've gotten in North Dakota, we're actually doing extremely well. Now, this is also a little bit misleading because breeding, that's what this line is. Breeding efforts started to increase in about 1965, 67. So if you only look at the data from that time, they do have higher gains in Illinois, but still we are above and beyond the capabilities of some other states. my concern is, and I didn't include this, is that now, so this was 2020, now we're kind of stuck at this 55 bushel per acre in the breeding program. We're kind of stuck at this 55 bushel per acre yield. We need to push past this because here I had, I apologize. This yield data was in kilograms per hectare and I cannot do that conversion in my head. So I gave it to you in bushels per acre. So in 2005, their yields were 60 bushels per acre. And now by 2020, they're up to 70. So they are still in the central Midwest, they're still seeing continual yield gains, whereas we have gotten to a point of, we're a little bit of a plateau. So it's important to recognize it now and say, what do we need to pass through to get through these plateaus and keep increasing our yield gains? Because I want 70 bushels per acre, right? Who doesn't? So let's look at this. This is, every breeder probably has a favorite equation, and this one is mine. This is the breeder's equation. You can write it a trillion different ways, but this is the format that I like. You have your genetic gains by year, by your selection intensity, your selection accuracy, which you might be like, what's the difference? One is how well do you select your parents? One is how well do you select your progeny? Genetic variance, how diverse are the materials that you're using? If you're only mating sibs by sibs by sibs, your genetic diversity is lower, which means your numerator is smaller, which means your genetic gains are going to not increase. And then you take all of this and divide it by time, as always, right? So each one of these numerators, you want to increase. You want to select better parents. You want to select better progeny. You want to increase the genetic variance, the genetic diversity in your program, and you want to do it fast, right? So actually the project that Clayton is talking about talks about both of these. And we are working in that same SoyGen project. So, but that's not for today's talk. So I am interested in this genetic variance. How can I increase genetic variance in order to increase my genetic gains? Which another way is saying, what can I do to get more yield? So the goals that I have are, one, let's determine the genetic diversity that we actually have. Find out if there's any bottlenecks. So one major bottleneck that's been identified just in all of North America is that, what is it, like 95% of all the genetic diversity in North America comes from 34 original plant introduction lines. 34. Can you imagine? I mean, I can't because I looked at my pedigrees. So, so if I understand this, where are bottlenecks? Where are places that we can open up genetic diversity? And then also with my background knowledge in traits that I know contribute to yield, how can we exploit them? Stem architecture, maturity, and then just getting new germplasm from areas that I already know are successful. So this work is going to be done by Forrest Hansen, a master's student in my lab. Technically, he's still an undergrad, but in May he starts as a master's student. So he has a couple more months of like easy life, and then things get serious. Actually, he's already done a good job. He already did all the pedigrees for those cultivars. So we went through all the cultivars, which is actually really tedious work. So we went through and wrote down all the cultivars, all the pedigrees of our commodity. Yes, of our commodity cultivars. I also am curious to compare the NDSU program to the founder lines. How we are in a unique environment. So how many of these founder lines actually contributed to our maturity group 0, maturity group 00 program? What if it's only like 4? Can you imagine? There's already only 34 lines that contribute to North America. And what if ours is only a handful of those? conduct PCA of principal component analysis of NDSU lines versus germplasm collection. This will give us a sense of how diverse. So the germplasm collection is essentially a collection of all the soybean that we would have access to in North America. So how different are all of our lines versus the germplasm collection? I want to determine linkage disequilibrium decay of NDSU lines. So have we lost diversity? Have we lost the chance for recombination over time in this breeding program, which could happen if you do too many inbreeds of closely related relatives? Determine nucleotide diversity. This is just a metric for genetic diversity. And then analyze key trait alleles present in the NDSU program. Thank you, AgriFlex. You made this one much, much easier. So since this is the master's student's project, but he hasn't actually started yet, so either my talk was going to end there or I can just do some work and get you some preliminary data. So this is a PCA. The genotype data is created for the AgriFlex marker panel. This is PCA of the 4 different projects in my program. So the 2 commodity groups, natto and tofu. Interestingly, or maybe not, because we already heard Nick's talk, the natto lines completely separate from the rest of the program. And I'm not surprised to see this because we already know that there's super rare alleles in that germplasm. So I assumed that the natto lines were not going to be similar to the rest of my program. So tofu's in yellow, natto is in, oh wait, not in, glyphosate tolerant is in purple and conventional is in blue. So the conventional program is really spaced out, which makes me think that there's relative, right? Because the PCA is only gonna be relative to the data that you put in. So relative to these other projects, the conventional program seems to have the most genetic diversity. I'm very surprised to see the tofu is pretty well embedded in there. But I also, like I said, glyphosate tolerant is my most commercially successful project. But if you observe how clumped together it is, this indicates to me a potential bottleneck, that there isn't much diversity here and that we might have gotten to a point where we're now crossing SIBs by SIBs. And so if I want to keep this program thriving, I need to start incorporating more of my conventional lines. Next, I compared my program to the founder lines. So those 34 lines that I talked about, I got the list from this really nice, really well done paper by Gislace et al. And so the founder lines are in this purple, and then glyphosate, again, now the natto go even further, right? Because there's definitely nothing like the founder lines. Glyphosate tolerant is in pink and conventional is in blue. And so you can see that there's a large portion of the founders that don't influence at least North Dakota germplasm. If you know anything about the founders, you're not going to be surprised to see Lincoln right there, right there in the middle of it. And then if I look at this whole area around that surrounds my program, these are the list of founders that I see. And you know, some people might be like, where's Harrisoy? Where's Clark? Well, those are actually progeny of the founders. So Harrisoy is AK, oh geez, wait. AK Harrow by Mandarin Ottawa is Harrisoy. So anyway, but this list is what I expect are going to be our contributors from the founders. So I said we already started going through some pedigree analysis. So there you go. Everyone's got that, right? I'm just kidding, obviously. So these are actually the pedigrees for 2 release varieties because Council is a parent of Ashtabula. And so here, you know, I just simplistically, the ones in blue are unique PIs that are not considered founders. So that's nice to see some diversity, but all these ones in green are founder lines. And you can see they're not very removed there hasn't been a lot of recombination or a lot of combination opportunities because these lines are so young. So the most predominant is Lincoln, and then Richland, and then Harris Soy. Oh, I was right. Mandarin Ottawa by A.K. Harrow. So these are the most predominant. I think Richland by Lincoln is Clark. Thank you. So not surprised, Clark, Harris Soy are the main contributors to this maturity group. What I think is also interesting is that at one point, pioneer lines got introduced, and they even have the same problem. It's all founders, and they're all the same. Lincoln, Richland, Lincoln, Richland. Even the pioneer lines in this maturity group are extremely related. So right here, I'm like, there's an opportunity. We just need, even if we bring in lines that are progeny of other founders, that's already an opportunity to increase genetic diversity. So essentially, my future research summary: identify bottlenecks, which I can just from the preliminary analysis, seems like there are, and find areas for germplasm improvements. So to do this, I can use pedigree records and genetic analysis. And then I really am looking forward to exploring maturity and stem architecture. I will most likely have to use historical data for that, just because since I've been here, the maturity data is not very consistent with early frosts and the drought that we had. And then I aim to increase genetic variance by breeding lines from different maturity groups. And then eventually what I want to do is do association studies with those to hopefully nail down some kind of vital QTL or chromosomal area that's missing from North Dakota that could, well, hopefully instantly boost yield gains. I mean, and that's what I hope for. I'm always like swinging for home runs. So that's what I hope to see. Most importantly, I need to acknowledge my breeding team. Ashley Cooper is our lead technician. Vicki Magnuson is our molecular biology whiz. Aaron Fromke, weeds guy, awesome, very important. We have Brock Fagerstrom, Evan, he's our mic runner. And Forrest Hansen is the master's student who's joining our lab. If you know me, I ask a lot of questions. So I'm really grateful for the people who take the time to answer my questions. So at NDSU, those people would be Juan Arsono, Drybean, Phil McLean, Bioinformatics, and Nanoy Vandillo, Pulse. Again, US soy breeders, are really grateful to them. Ted Helms, of course, this work would completely be impossible without him. And Randy Nelson, both of these guys have retired. Aaron Laurent at University of Minnesota, and Andrew Skiboo and Brian Deers, especially because they've been mentoring since my PhD and they haven't told me, leave me alone yet. So I'm really appreciative for them. And then of course, University of Missouri, Kristen Blue was my PhD advisor, still my mentor. Nick Dietz. Anytime you see a graphic that I show, Nick Dietz probably helped me troubleshoot it. I would not know ggplot without Nick. So I will always be appreciative of you. Funding, Soybean Council, of course, always indebted to them. And now branching out funding to NCSRP. And of course, NDSU has been unbelievably supportive of me and my breeding program. So with that, I can take any questions. Oh, and it was requested on Zoom that if you ask a question to wait for the microphone because they can't hear us. So.
Well, thank you, Carrie. That's pretty cool. Very nice stuff going on. I was just wondering on the diversity. I agree with you. We really have not explored much of the diversity available. I was wondering, what do you expect from incorporating those ancestors in your program? Especially based on adaptability, I would imagine the ones that you have on your pedigree are really adapted here. The second one is when you're showing that the PCA, have you tried to look over time? Like how did your releases change over time related to the ancestors instead of looking just at like a one-time thing?
That actually is interesting. No, I hadn't thought about that. That would just depend, because what I showed there were I didn't explain it well, actually. I forgot to mention that also includes not only the cultivars, but my current advanced yield lines. So in order to do that kind of work, which would be very cool, I would need to have genetic access, or like seed access to historical lines, which I think might actually, like from the crossing blocks, we usually save the parents. So that is an interesting thing to explore. So your first part of the question, when you're asking about the breeding effort for germplasm diversity, yeah, it's a challenge. A lot of people don't do it. You know what I have to do? I already have some materials from Illinois, maturity group 2, and then I will soon be getting some from Nebraska, because they are the ones that have the highest yielding lines in public programs, 70 bushels per acre. So of course I'm hoping, cross by 70 bushels per acre. Hopefully I'll even get an average of that of 60. That would be great. But you know what I have to do? I have to grow them in growth chambers. I have to regulate the day length in order to trick the plant to flower at the same time as the North Dakota lines flower. And then once I do that, you would think, I'm done. Because at first I was, I'll be done. And then I was like, wait, no. Because then it's just an F1. So then it's 50-50, right? I can't, it's not going to work. So essentially I need to grow these in growth chambers, or at least in a winter nursery, for probably about 3 or 4 seasons before I can bring them up here and then select for our environment. So it's certainly effort, but if that effort can get me a 10 bushel per acre yield gain, which is again what which is what I'm hoping for. And then I also have a mapping population with that too. So that's where my thought is. Start there at least. Yeah, thank you. I did that well of a job. Come on.
So there is an interesting kind of discussion about, you say my soybeans yield 55 bushel, whatever. But if you look at the farmers, what they get, on average 35, if you get
That's
last
right.
year. So between what the farmers get and what you are thinking the potential is, I'm sure the potential is higher, because if you grow some of your varieties in really good conditions, it gets more than what you're presenting there.
Yeah, that's why I only show dry land.
Yeah, but still, what I'm talking about is that you're way ahead of where the farmers are. So, yes, we want to get your numbers up, but there is time to also increase what the farmers can obtain from the genetics that is already there.
Yeah, and this is a trend that we see in all the breeding programs. The breeding programs get the highest yields, and then farmers usually get about 10 or 15 bushels per acre less. And so that usually is because our plots are, we have 100 acres in our breeding program, but still they get a lot of attention, right? If I see a weed, Aaron, can you guys go out there and hoe? A farmer's not doing that on 10,000 acres. And so, weed management period for a farmer is much more complicated than it is for us. And that's why we see higher yields, even of the same genetic materials. And so, but essentially with the thought amongst breeders, because I've asked this question before, the thought amongst breeders is that it's our job to maximize that genetic potential. So even though we know that there's going to be a bit of a yield loss when it gets, scalability, once it goes to large scale, we know that there's going to be a bit of a yield loss. But if we can still increase that potential for the farmer, they should still be able to go from 40 bushels an acre, hopefully to 50, 45 even. That's a significant difference in money when you look at it over thousands of acres. So yeah, it is a discrepancy that we're aware of. Yes, Evan. I was going to say that too. That's your job.
So Carrie, you're kind of in a unique situation as a breeder in North Dakota, because you also deal with some extremes, the northern coldness and also the western dryness. And I guess the question is, in your program, how do you add those things to your breeding program that you have.
Yeah, and this is something I've been thinking about since I first got here. I mean, that's why, you know, I've been here a year and a half, and now I'm only introducing the concept of my research because I've been trying to understand where's Williston, you know, where's Minot. So now that I'm getting more comfortable understanding the state and its environment, I do understand actually Hans has a really nice map that you show the maturity. The maturity bands are not linear like you see them by latitude in other states. Ours are more like this, right? Because of the dryness out west. And so so this is more general in terms of research for yield gains. But when it comes to the specific environments, especially out west, I am looking into ways to ameliorate, or no, I should say, to enhance soybean growth in low water conditions. And I think that out west, who is one of the biggest, well, they're the ones that are buying all the NDSU soybean. So they're some of our best customers. Their needs are very important, and their biggest need is how to get maximal yield in low water conditions. So the background, the high yield potential background is still useful, but then I'm also looking at other traits that could help them in low water conditions. And then, yes, and then on top of it too, I know in the north, I need really early lines, right? 0.06, 0.7, which is really unique. Although, I mean, look at GT. Oh, I forgot to tell you guys. Yeah, if you look on the backs of your hats, they all have the name of a soybean variety, which was featured in my research. So this one is 1406 high protein. If you got the 17009 GT, that's our moneymaker. So good for you. Yeah, there you go. So, I mean, that's why trials, you know, the RECs, the trials that we do at the extension centers are invaluable because, and the advice that I get from you guys too, right? I'm not going to go to Brian Hansen and tell him what variety he needs to see in his environment. I'm going to say, Brian, tell me which one you like. So, yeah, working with the RECs is invaluable for releasing varieties specific to different environments in the States. So, all right, I went way over time. I'm really sorry.